Hi there. I'm here to present a project I was able to do over the summer at the UO thanks to the generosity of CURE, specifically through the FYRE Award for first years. And the project I was able to do through the Sylwestrak lab is called The Role of Lateral Habenula Tac1 Neurons in Reward Prediction Error, or RPE, Responses to Conditioned Reward Predictive Stimuli. So I'll talk a little bit about the experimental design process. That was one of my favorite parts. So just a bit of background, I'll move this, about what RPE is and the lateral habenula. So RPE stands for reward prediction error. As for the diagram on the right, you can kind of just think of it as the difference between what you expect an outcome to be in terms of a rewarding outcome and what the actual outcome is. So positive reward prediction error is a better than expected outcome, where negative reward prediction error is a worse than expected outcome. And so there are some systems in the brain that are activated by positive RPE, such as the midbrain dopaminergic system, which is a more commonly understood one, where essentially some better than expected outcome happens pertaining to reward, and there is an increase in dopamine firing. But the lateral habenula is a region that is activated specifically by negative reward prediction error. So you'll see an increase in activity, like a pretty transient increase following a worse than expected outcome. And this, whoa, sorry. Okay, so the lateral habenula is involved in a lot of different negative signaling. So that might be fear, anxiety, stress responses, but that includes negative reward prediction error. However, because those systems have a lot of interplay, it's hard to sort of isolate and study reward processing in the lateral habenula if you're recording from the entire region because of all of those other phenomena going on at the same time. So the lab that I work in, the Sylwestrak lab, has previously identified tachykinin 1 expressing lateral habenula neurons. So we call those lateral habenula or LHB Tac1 neurons. Identified those as a potential subpopulation that's specific to RPE, in that it will respond to situations that evoke negative reward prediction error, but it's not responsive to other reward unrelated aversive conditions. So they were able to find this by conditioning mice to expect a reward when they poke into a certain port after performing an action. But 10% of the time there would be no reward, and you would see this like pretty transient increase in neural activity from these Tac1 neurons specifically. Whereas whenever they would do some aversive stimuli, like an air puff to the face, there would not be a response from these neurons in the same way. So to developing the research question, we want to look at this potential group of neurons, these Tac1 neurons, right, and see, do they show other traits of RPE? And are they the only or the main subpopulation in the lateral habenula that's responsible for reward encoding? So RPE, it's I guess, a well-accepted phenomenon that if you have a predictive cue and then your outcome, your reward outcome, then the reward prediction error response will shift from the time of the outcome to the time of the predictive cue, because that's essentially when the information of what's going to happen is updating. And so that's been observed in the lateral habenula before. But since we have this evidence, that leads us to consider the idea that these Tac1 neurons are specific to RPE, we would like to record from Tac1 neurons and then implement this Pavlovian conditioning style task to see whether the Tac1 neurons are responding to the predictive cue, in the case of like a low probability predicted cue, because that would be the more negative reward prediction error outcome. So if they do, we would be led to assume that, or it would just back up the idea that these Tac1 neurons are specifically associated with reward prediction error. But if not, it would maybe tell us that there's at least like another subpopulation of neurons involved in reward prediction error that's specifically responding to this predictive cue. And both of those are interesting. So our strategy for looking at this was, we had this Pavlovian conditioning setup with a reward port, from which we dispensed sucrose water. That was the reward. All these things are common in the literature. We had a beam break in there for detecting when they entered into the port, and that was how we registered or marked a timestamp for the onset of the reward outcome, so that we could align it to their neural activity. And then Yeah, so we had two auditory cues of different frequencies counterbalanced. So half of the mice had, or they had opposite contingencies just to account for maybe if the frequencies elicited any responses themselves. And then, yeah, so the sucrose water would be dispensed or emitted, and after the cue is played, so that anytime that they come up to the cue port, there either will be a reward or won't. And as for recording specifically from these Tac1 neurons, we use a strategy called fiber photometry, and we also use like a Cree system to only target Tac1 neurons. So that essentially, like, we have transgenic mice in which any genes or any neurons that express Tac1 will also express Cree. And then we have this Cree-dependent calcium sensor where in these Tac1 neurons, because they're expressing Cree, when they're activated or when they're passing on an action potential, they'll intake calcium, and that will cause these neurons to fluoresce. And so we have a fiber implant that sits over the lateral habenula that will pick up the population fluorescence levels, and that's our proxy for neural activity. But we're able to record it in vivo while they're running through the task. So yeah, in the process of running this experiment, we had a lot of troubleshooting to do, a lot of like conceptual limitations in that we had some technical issues, but we had to think about what ways we could solve them without ruining the integrity of the experiment. I will say that we had a lot of troubleshooting to do, and so the integrity of the experiment at that point is not the best, but it was a very big learning experience, this whole thing, and I came out of it having developed a much more stable way to run the experiment than how we had started, because it was just new to our lab to have this type of auditory cue Pavlovian task. But yeah, we'll talk a little about results. Just to address the research question, which actually wasn't even the bulk of everything that we learned when looking at the data. The question being like how or if or do these lateral habenula Tac1 neurons respond to these reward predictive auditory cues in this Pavlovian setup. And so we expected, because we've seen them respond to reward emissions, which are a form of negative reward prediction error, we should see a similar increase in activity following the low reward probability predictive cue, because it's essentially updating them that they're having a really low shot at getting a reward, which could be considered a worse than expected outcome. So this figure is probably the most confusing figure that I have. What you're looking at is a heat map. This is essentially tracking the fluorescence levels, and the coloring is z-scores, or actually DF values, which is specific to this fiber photometry, DF values away from the baseline. So essentially, the higher the activity level is compared to the baseline, the more yellow it is on this. And so I will say, like, each of these rows is one animal for one session. And so the activity that you see aligned to the reward onset, which is the whole second column, RE1, that gray line is the reward onset. Everything after it that's like time locked to that, you're also seeing in the Q1, because the onset of the cue and when they go and get the reward is usually about like a 1 second time latency. And so the activity you're seeing 1 second after the cue onset in the left column is more likely the activity from the reward onset. So that makes it difficult to look and see, is there activity aligned to the cue onset? But there definitely is not any strong pattern within, like a 0.2 seconds of the cue onset, the way that there is with the reward onset, which is part of our conclusion that cue responses are not being observed in recording from these Tac1 neurons. So we have yet to do statistical analysis to quantitatively compare those changes, but there is a clear difference just by visuals in terms of where a response starts in relation to the cue onset or the reward onset. And yeah, so that figure can be a little confusing because you are seeing these blocks of activity after the cue onset, but the idea that we're putting forth is that they're likely the same as these ones, but just 1 second after the cue, because that's where the reward onset is, and the time window is a lot bigger. Yeah, and so then our observation, though, was just that these Tac1 neurons don't show an increased like phasic, or not phasic, but transient response, just like a sharp increase following the predictive cue onset, which suggests like they might not be involved in predictive response, or the tasks just didn't really evoke the responses that well. But we have another way to kind of evaluate how well these predictive cues were actually providing information. And that's by looking at the responses to the reward outcomes themselves. And so we know that Tac1 neurons respond to reward emission, which is at the time of the outcome. So we wanted to see whether the magnitude of these responses is affected by, I guess, the amount of error. So whether you have like a low or high probability predicting cue and then it's emitted. If it's a high probability predicting cue, it's a lot bigger of an error than if it's a low probability cue and then emitted. So if we see a strong correlation, then we would be more inclined to believe that the auditory cues are somewhat being registered by the animals and that they're providing information. But if there's no correlation, we would be more inclined to think that the setup is not evoking or is not allowing these predictive cues to really serve their purpose. But this is a representative day, but I promise like they really did follow this trend from day to day throughout training. And it's looking at the low probability condition met with like the emission outcome, and then the high probability cue with the emission outcome. And these are the delivered with the same probabilities respectively. So essentially what we're seeing here is that there's a huge increase in activity comparatively following the high probability cue and then the emission response. So these are time locked to the reward outcome, but it's providing context of what cue was played first. So this suggests that after the high probability cue is played, and then there's a reward emission, which is the largest, I guess, magnitude of negative reward prediction error that all of these four possibilities can create. You see this largest response, and you see some response for the low probability cue and then the emission following it, as shown to the left here. So this is also looking at reversal. So we also switched the cue contingencies for each individual animal. So what we're looking at here that says P low emission is the now low probability cue and then the emission following it. But it's the original high probability. And so if it's day one of cue reversal before this like learning has, I guess, happened, it looks more similar to the original P high emission pattern. Now this is the old P high emission and the new P low. And so similarly, the original P low, or like low probability to emission situation, we didn't see much responses like this. And so that's reflected in this day one pre-training. But through 9 days of cue reversal, you sort of see this switch where what is this low probability, like this new low probability cue is actually, you're losing some of that like magnitude of the response as the association is built that this is now the low probability cue, whereas this newly defined high probability cue, you gain this response as the association is built that this cue predicts a high chance of reward. Then when there's an emission, there's a bigger response. So that kind of reflects learning and reward prediction, which is interesting because we've not really looked at learning process before, but it was really cool to see. And so our interpretation from that was just that like what we gathered was despite switching the cue contingencies, we still see that pattern of a larger response in the like high probability cue to emission situation. And we're still talking about like at the onset of the reward outcome, but just giving the context of the cue. So that correlates with the idea that like a larger magnitude negative reward prediction error elicits a larger response or a larger magnitude response respectively. And yeah, so we also did see that like in the low probability cue and the emission following that, there was almost no response, right? We can look at this one, it's easier to look at. There's almost no response, even though there's an emission, but we know that from previous experience that Tac1 cells reliably respond to emission. So there's this idea that because they had a low shot of getting rewarded and that there's a low probability, perchance that's like lowering the amount of RPE that they're experiencing. And so it's decreasing the magnitude of the response. And so, yeah, so that gives us some idea that potentially these cues were at least being associated with their respective contingencies. So yeah, to think about data integrity, we had to make a lot of changes throughout the experiment just to make sure that the task setup was functional. And I would not feel confident making conclusions off of this data because of that, because the methods were inconsistent. However, we were able to come up with a stronger method plan through this experience, which I think was fully worth it. We're still in the progress of statistical analysis of these net fluorescence data, which would be awesome to really make strong comparisons though. And we have to verify the implant and injection location. So in terms of what neurons we were recording from and where, we can look at those through histology just to see how accurate that was. So we could know if we were recording from some other region or if expression levels were really high or low. And yeah, we had a little bit of a issue with the reward port with, I guess, registering accurately when the animal becomes aware whether there's reward or not. It's a bit hard to decide that just by like where our beam break is placed and how far they enter into the port, but that's something that we're developing and continuing for the future project. And so yeah, our future experience, we want to look at comparing, it's essentially we want to do a similar project but with a more refined and consistent methods, but also comparing Tac1 and non-Tac1 populations in the lateral habenula, because that will give us a better idea if RPE responses are being seen to predictive cues at all. Because in the literature, there are RPE responses to the predictive cues in the lateral habenula, but we didn't observe them, but that could be because the experimental setup wasn't good to evoke them, or it could be because the Tac1 neurons are not responsible for that. So this would give us just a better control idea of what's really happening. And just a thank you to Cure for the funding, and to Lanch and Kevin and Brian and JC for all of the guidance and support that I've been able to receive over this summer. This project has been amazing to do, and a special thanks to Dr. Sylwestrak and everybody in the Sylwestrak lab, especially Kana, for just amazing mentorship and guidance all along the way. And thank you for watching my presentation.